Welcome back to our ASCP Story series. In this video, we are tackling another set of challenges for you. Our first question is about the messenger of our immune and hematopoietic systems, cytokines. Interleukins and colony-stimulating factors are cytokines produced by C. Monocytes and T. lymphocytes. Cytokines are a broad category of small proteins that are crucial for cell signaling. They act like commands telling other cells what to do. Interleukins, ILs, are a type of cytokines that primarily mediate communication between white blood cells. Colony-stimulating factors CSF are cytokines that tell hematopoietic stem cells in their bone marrow to grow and differentiate into specific types of blood cells. T lymphocytes, especially helper T cells, and monocytes, which mature into macrophages, are the master regulators of the immune response. A huge part of their job is to produce a wide array of cytokines. Next up is the fundamental physiology questions. What is the appropriate total blood volume in a death? The correct answer is C, 6 liters. Which of the following factors will result in an immediate increase in oxygen delivery to the tissues? The question is about the oxygen hemoglobin dissociation curve. The answer is C, increased hemoglobin binding of 2,3-PPG. 2,3-PPG, also called 2,3-DPG, is a molecule inside red cells that acts as a regulator. When 2,3-PPG binds to hemoglobin, it stabilizes the T or tense state, which has a lower affinity for oxygen. This forces hemoglobin to release its oxygen. So, an increased 2,3-PPG binding causes the right shift and immediate increase their oxygen delivery. Increased pH, alkalosis. This causes to the left shift. Hemoglobin holds onto the oxygen more tightly, decreasing delivery to tissues. High altitudes. This is a stimulus for adaptation, but the immediate effect is hypoxia. The body responds to high altitude by increasing 2,3-PPG, but high altitude itself is the direct mechanism. Increased renal release of erythropoietin EPO. This is a long-term adaptation. EPO stimulates the bone marrow to make more red cells, which increase the total oxygen carrying capacity over days to weeks, not immediately. The fatty nuclear maturation commonly results in the production of red cells that are C, macrocyted. This question links a specific cellular defect to red blood cells morphology. The fatty nuclear maturation is the hallmark of megaloblastic anemia. The essential components for DNA synthesis and cell division are vitamin B12 and folic acids. When one of these is deficient, the cell nuclei cannot mature or divide properly. However, the cytoplasm, which doesn't need B12 or folate to make hemoglobin, continues to grow. This results in the production of abnormally large red cells known as macrocytes. What is the most common cause of iron deficiency? Here's a question about the most common anemia worldwide. The answer is A. Bleeding. In adults, the number one cause of iron deficiency is chronic blood loss. The body is very good at recycling iron, so it's hard to become deficient unless you are actively losing it. Common sources of chronic bleeding include gastrointestinal, GI bleeding from ulcers, tumors, etc. Heavy menstrual bleeding in premenopausal women. In the anemia of chronic disease, what are the usual serum iron and transferrin levels? This is a key question for differentiating types of anemia using iron studies. The answer is A. Serum iron decrease, transferrin decrease. Anemia of chronic disease, ACD, also called anemia of inflammation, is driven by the hormone hepcidin. During chronic inflammation or infection, the body releases hepcidin, which does two things. The first one is it blocks the absorption of iron from the gut. And the second is it traps iron inside macrophage, preventing it from being used for erythropoiesis. This is a defense mechanism to keep iron away from invading pathogens. In children, the most important effect of lead poisoning is on the C. Neurologic system. Lead is a toxic heavy metal that can affect multiple organ systems. It is particularly dangerous for children because their bodies and brains are still developing. Lead exposure can cause a lower IQ, learning disabilities, behavior problems, and developmental delays. This neurotoxicity is the primary reason for public health efforts to eliminate lead from the environment. Which of the following values can be used to indicate the presence of hemolytic anemia? The answer is D. Reticulocyte count. Reticulocyte count measures the number of new general blood cells being released from the bone marrow. In hemolytic anemia, red cells are being destroyed in the periphery, but the bone marrow is usually healthy. To compensate for the destruction, a healthy marrow will ramp up production, releasing a high number of reticulocytes. A high reticulocyte count in the face of anemia is the hallmark of compensatory response to either hemolysis or acute blood loss. Our final question is a case study that requires you to put all the clues together. Patient, 20-year-old female. CBC results, mild microcystic anemia. It means low MCV. Smear, target cells and stable red cells, basophilic stippling. These are classic but non-specific findings in disorder or hemoglobin synthesis. The key clue: hemoglobin A2 level 5%. Normally, hemoglobin A2 makes up only about 2.5 of total hemoglobin. So the answer is C. Heterozygous beta thalassemia. In heterozygous beta thalassemia, also called beta thalassemia minor or trait, there is a defect in one of the two beta globin genes. This leads to reduced production of beta chains. To compensate, the red cell precursor increased the production of delta chains. These extra delta chains combine with the available alpha chains to form more hemoglobin A2. An elevated hemoglobin A2 level, typically 4 to 8%, is the definitive diagnostic feature of beta thalassemia trait. I hope you learned something new. Keep up the great work and see you in the next video.